is with great pleasure that I introduce Nikos Logothetis, who will be presenting our STOR Major Issues in Biology seminar today. Nikos received his BS in Mathematics from the University of Athens and a BS in Biology from the University of Thessaloniki, and a PhD in Human Neurobiology from the Ludwig Maximilians University in Munich. He was a postdoc and professor in the Department of Brain and Cognitive Science at MIT, and then joined the faculty at Baylor College of Medicine in Texas. Nikos is now a professor and the director of the Max Planck Institute for Biological Cybernetics in Tübingen, Germany. It would take more time than we have to summarize the numerous scientific advisory boards and honors that Nikos has received, so I mention only a few. Nikos is a member of the German Academy for Sciences and a foreign associate of the National Academy of Sciences. He is a recipient of the DeBakey Award for Excellence in Science, the Golden Brain Award of the Minerva Foundation, the 2003 Louis Gintet Prize of Medicine, the 2004 Zolch Prize Award for Neuroscience, the 2007 Ispin Prize for Neuronal Plasticity, and the 2008 Alden Spencer Award of Columbia University. It would be impossible to summarize all of the substantial yet eclectic contributions Nikos has made to the field of neuroscience and biology as a whole, in part because his contributions span so many sub-disciplines, including neuroengineering, computational biology, visual psychophysics, cognitive neuroscience, in vitro electrophysiology, scalp EEG, neuroanatomical mapping of the visual system, and the neural basis of social cognition. Perhaps a personal anecdote will best convey the impact that Nikos has had on my life and that of my colleagues, because I can't begin to summarize all of his contributions in 3 minutes. When Nikos' results in 2001, showing that the fMRI bold signal, the dominant method in cognitive neuroscience for assessing behavior-brain interactions in humans, was first published, his results were met by an interesting combination of excitement, confusion, extensive debate, and of course, heated discussion. Suddenly, Nikos' results and name had become a household dinner and student-faculty drinking topic, with almost all wondering how he had managed to simultaneously record fMRI and electrophysiological activity. In his seminal and now well-known work, Nikos showed that the poorly understood yet well-known fMRI bold signal did not consistently represent single neuron activity that we have all come to know and love, but instead represented the aggregate neural signal called the local field potential, far more mysterious and far less well-known. Perhaps as a sign of truly groundbreaking work, in addition to the flurry and excitement surrounding this work, some wondered openly about the future of the entire methodology of fMRI. Fortunately for us, following his work in 2001, Nikos published several review papers putting his work in context and explaining that, in fact, all was not lost for us in cognitive neuroscience. Thank you for that. Nikos continues to do groundbreaking work, and I will mention only a selected few of his recent contributions, including explaining in mathematical detail what the local field potential means and the neural basis of scalp EEG in visual cortex. Nikos will discuss electrical stimulation in fMRI today, yet another novel and highly relevant topic to neuroscience. We are very fortunate to have Nikos as our guest speaker today. Let's please give him a very warm welcome.
Okay. Many thanks for this kind introduction, and my sincere thanks also go to the Ruth Storer Endowment Committee, which elected me as a lecturer for the Major Issues in Modern Biology series. Now, before I start, a few comments. If I start speaking Greek, wave your hand. It's my first talk that I give 1 hour after midnight, so it's an interesting experience for me. Questions, just raise the hand and ask the questions. I don't need to go all the way to the end of the talk if there are things that we should clarify in a short discussion. Thank you. Now, my talk today will be on electrical stimulation of the brain combined with intracortical recordings and fMRI. I call it ES fMRI. Scientists have been applying electrical stimulation to animals and humans for over 100 years now. And although some might say it is a crude method for understanding detailed mechanisms underlying various neural computations, microstimulation has undoubtedly made significant contributions to our knowledge in both basic and clinical research. Recently, there has been resurgence in its use in the context of electrotherapy and prosthesis, as you may know. Nonetheless, as you may also know, the methodology still suffers from many fundamental problems. James Rang got right to the point in his excellent review in 1975, saying that electrical stimulation of, say, the lateral hypothalamus is a shorter version of the statement that there was a stimulating electrode in the lateral hypothalamus which affected an unknown number and unknown kinds of cells at unknown locations in the vicinity of the electrode. Well, I must admit, we still do not know exactly what's being stimulated when we pass current through the tissue, and we still do not know how the signals elicited by electrical stimulation propagate in the brain. In fact, if we knew, I wouldn't be giving this particular talk today. A profound understanding of such issues is now more than ever necessary, and not only because of its usefulness for the clinic, which I must admit is not exactly my concern. So electrical stimulation together with fMRI promises actually to be an excellent tool for the study of projective fields and of effective connectivity and plasticity, as well as for spatially resolved mapping of any networks underlying electrostimulation-induced behaviors. And you might know that electrostimulation has always effect, but you may also know that we don't really understand why the effect is there and what it means when the effect is not there. So I believe analysis of neural activity at different spatiotemporal scales is now a must in systems neuroscience. Brains, you would agree with me, can be thought of as complex adaptive systems. They are governed by nonlinear coupling rules, and they do not lend themselves for easy formal modeling and simulation. Their components may themselves be complex systems, so that the notion of elementary operational unit depends entirely on the level of analysis. Unfortunately, neither the coupling rules nor the appropriate elementary units are really known. Let me illustrate this by using an example from the visual system, the primary focus of my own lab's research. The top drawing illustrates the retinogeniculus stride pathway of humans, and the bottom drawing shows the same thing in monkeys. Information flows from the eye, as you know, through the thalamus and all to the primary visual cortex in the occipital lobe. The primary visual cortex is also known as stride cortex or area V1, and it's the starting point of the cortical processing. A large number of cortical areas known as extrastriate cortex are devoted to different types of visual processing. Almost all of them have been identified in the monkey on the basis of physiological, histological, and lesion studies. On the top right, you can see these areas that are on the surface of the brain, and here you can see those that are hidden in the sulci. These types of latent representations are used also in human neuroimaging for the localization of activity related to a variety of cognitive tasks. Obviously, at a certain analysis level, such areas themselves may be considered components or subsystems in visual processing. But of course, the areas are not homogeneous, and their interaction rules are not entirely known. Each one of them can be recursively segmented into increasingly smaller operational units. You're going to see here what I mean. A small volume that you see here, for example, of tissue of the stride cortex has strong tangential and in-depth subdivisions. Cells in different horizontal compartments respond to different types of stimuli, as you can see here. And at the same time, cells in different layers may have different properties and are affected differentially from neuromodulation. So these cortical columns or their subdivisions could also be considered as ideal elementary operational units at the systems level, but even in the primary sensory cortical areas, there is a critical mismatch between ontogenetically defined columns and anatomically physiologically defined microcircuits like the one you see here. I'll come back to the operation of the circuits during the discussion of the microstimulation results. In a nutshell, it may seem convenient to concentrate either on the large spots mapped in neuroimaging or to relate changes in the firing of single units to the behavior of an entire organism. But real progress in the study of the neural mechanisms of behavior will probably hinge on a deeper understanding of information processing on different spatial scales, and also on a better comprehension of effective dynamic connectivity. And that's another word that is puzzling. I'll tell you exactly, I'll show you what I mean. On the left side, you see the massive connectivity diagram that was published by Ferriman and Van Essen in 1991. Properties at each level are strongly modulated by all other levels. This is complicated enough, of course, but actually there is more. The diagram here implicitly suggests that cortical processing of sensory events is purely an intracortical mechanism. Yet associative thalamic nuclei, such as the pulvinar, as you all know, are hard to ignore when we are investigating visual network activity. The convoluted and compartmentalized cortex that you just saw in the previous slides is shrink-wrapped around the barrel-shaped thalamus, and the somewhat blurred local connectivity generates matching corticocortical and cortico-pulvinocortical connectivity patterns. There are maps also here, and a general observation is that indirect cortico-pulvinocortical circuits tend to mimic the direct corticocortical pathways. If two cortical areas communicate directly, they are also likely to communicate through the pulvinar. I have taken these few minutes to briefly mention some important conceptual issues because all developments and investigations in my lab over the last decade have been driven by the conviction that it's now essential to consider these issues in future experimental plans. So basically, what we have done is we started working not only in continuing the research we have been doing all these years, but also developing technologies. And among the things that we paid attention to was, first of all, the interpretation of neural signals, which is a big issue, is a better understanding of the MR neuroimaging signals and the development of new MR molecular imaging, like the one that you see here, work on neuromodulation, see the effects on cortical processing, but also on the neurovascular coupling, and finally, in vivo connectivity.
Well, we could if we knew how to do it, yeah.
Is it better now?
There's an interference with the frequency in that they have set inside that media closet.
Well, the mic is off. Right now. Has been. Want to try it again?
Yes, can you hear me now? Did you hear anything from what I was saying? Let me repeat it. Okay. Anyway, so the connectivity is important, as I said, because it's the only way basically that you can link different states of analysis. And the classical standard connectivity was done with degeneration studies with conventional traces and transneuronal traces. And there is no substitute for that. It's of extremely high resolution, wonderful method. But as you know, it requires fixed tissue. Can you hear me now? Or
Yes.
Okay. It requires fixed tissue, and it cannot be used in longitudinal experiments. So there are alternative things. For example, there are all this diffusion tensor imaging and effective connectivity that relies on modeling. And there are also some invasive techniques that have high resolution because the DTI, although it's an interesting method for very rough description, basically, of bundles, it's not something that you can study when you want to know cortical connectivity or any U fibers or any things that are close to cortex where the anisotropy, basically, of water diffusion is very small. The paramagnetic traces, on the other hand, they are very promising. As you can see here, if you use manganese and you inject it with intraocular injections, you see the visual system that I just described to you. You can see very well the optic nerve, the optic tract, and then through the optical radiation, basically, the manganese going to the cortex. So you can see up to 4 synapses. Basically, you can almost see the entire brain if you want. And combined with electrostimulation, it's a very good method to set the anatomical constraints and then study, basically, dynamic connectivity. Now, what I'm going to do today, I'll concentrate on electrical stimulation and fMRI, and this is basically the apparatus that we use for the experiments you're going to hear. On the left, you see a 4.7 Tesla 40 centimeter magnet, and on the right, you see the 7 Tesla 60 centimeter magnet. For the molecular imaging project and for certain types of anatomy in small animals, we use also the 16.4 Tesla and 20 centimeters magnet that can give you resolution up to 50 micrometers, actually, isotropic. And with parallel imaging, the resolution can be close to what you would get with an MR microscopy. Needless to say that we needed to do a lot of developments. I'm not going to go into any details because we don't have the time, but both hardware and software had to be developed from the beginning for the systems. The systems are almost custom made. The magnet and the consoles were initially made by the company, and the rest was taken over by my own laboratory. So the first things that we did is to develop our new coils and sensors and electrode holders, and of course interference compensation that you see here, all this published work. And on top of it, for the alert animal, we had to have transport systems, we have to have mock setups, and most importantly, we have to use all these sensors that can be used to train the animal on the basis of operant conditioning techniques. Nowadays, we can do the following things with the systems on a daily basis. High resolution anatomical MRI, 100 by 100 by 100 micrometers. High resolution MRI, 100 by 100 fMRI by 700 micrometers with implanted radiofrequency coils. Simultaneous intracortical recordings in fMRI, as you know, capillary microsampling connected to HPLC and tandem spectrometry, electrophysiology in fMRI, electrostimulation, intracortical recordings in fMRI, amperometric polarography, high resolution chemical shift imaging, spectroscopy, and of course pharmacological fMRI and the manganese enhanced MRI with osmotic pumps. Today I'll talk about two things. The first one is going to be on the stimulation of cortex. We collected data while electrically stimulating the primary visual cortex in the area MT of monkeys in order to characterize the activity patterns that are generated locally at the site of the stimulation and distally at regions innervated by the site of the stimulation. These initial studies indicated that electrical stimulation activates only monosynaptic targets. We'll discuss that thoroughly in this talk. To better understand this result, we followed up with experiments in which we stimulated the thalamic nuclei that provide input to the primary visual cortex and examined the physiological and fMRI responses in cortex. In the second series of experiments, we used simultaneous microstimulation, electrophysiology, and fMRI to unveil potential global changes in brain activity due to local hippocampal plasticity. As you will see, the findings provide the first evidence of LTP-induced network reorganization after changes in synaptic strength within the hippocampus. Now, this is just to remind you basically something about the organization of the primary visual cortex and the posterior visual areas, and also to show you that the mapping of distal activations, which can be evoked during V1 stimulation, benefits greatly from the retinotopic organization of all posterior visual areas. All these areas have a map of the contralateral field, as you know, and you have the fovea somewhere here, and this is something like 7 or 8 degrees. And the same map is also in area V2, V3, V4, MT, and so on and so forth. The boundaries of the areas here, for example, are shown by superimposing the drawing on the T1 weighted image. Now, the retinotopy predicts that if a small region of V1 is activated by electrical stimulation, distal activations should be found, first of all, in all retinotopical image regions in each one of the extrastriate areas. Here, the activation is induced by visual stimulation, and you see that it's a small stimulus presented at 2 degrees eccentricity is going to give an activation in the primary visual cortex here, and it's the match point in area V2. And if you increase the eccentricity of the stimulus, you'll see that both things are moving along V1 and V2, and here with the 4 degrees, basically, you have the 2 spots here that represent, again, the retinotopically matched locations in cortex. Not surprisingly, electrical stimulation of V1 yielded retinotopically matched activation. The upper left image here shows the location of the electrode, and the right shows basically the standard the standard maps, activation maps, and you see that stimulation of the cortex here induces activation in the posterior bank of the lunate, in the posterior bank of the STS, and these positions here, the way we did the retinotopic mapping initially, they are matching positions. On the bottom right, you see the time courses, and these time courses are basically indistinguishable from those that you obtain if you present a very small visual stimulus in the visual field. Now, an interesting observation, as I said, is that the activated areas that are summarized here were monosynaptic targets. Now, before we go into the details, and before we start investigating this and see whether it is true and what it means, we wanted to do two things that are essential for understanding what you're doing with microstimulation. We wanted to calculate the shape and size of the passive current spread, and also to calculate the excitability of the stimulated area. That means the physiological property that permits a rough estimation of what is excited during this stimulation experiment. Now, the bolt activity, as you see here, as expected also, spreads within the slice plane and across different slices. The three-dimensional reconstruction here shows the spread in the operculum from which the radius of the activation can be computed. Electrical stimulation evoked a bolt response that had a maximum spread of about 6 millimeters. Now, the radius typically increases with the square root of the current, as you can deduce from the simple equation here, and the constant K has been calculated by several investigators. If you use a value for something like 1.2 milliamps, yields the dashed line that you see. Do you see the dashed line? What happened? Oops. Okay, I'm sorry. The dashed line that you see here, and the dots show the data from our experiments. The bolt activation is about 2.5 millimeters larger than expected from the activation of pyramidal neurons, showing once again the sensitivity of the fMRI signal to subthreshold processes. The second property that was essential before we investigate all the phenomena that I'm going to be describing to you is the excitability. Now, such measurements rely on the fact that there is a trade-off between current intensity and duration of a pulse. If you start with a pulse duration of 400 microseconds and you keep increasing the current in microamps, then you're going to get a sigmoid, basically, curve. There is a plateau. At some point, you don't get more volume activated, no matter how much current you use. And if you reduce, basically, the duration of the pulse, there's going to be a shift to the right, which means you need, for each one of these volumes, more current. Now, this is a standard thing that has been done, and you can define a threshold. And the threshold current plotted against, basically, the pulse duration gives you the so-called strength duration curves that are extremely informative. They're not linear. They decrease and they reach an asymptotic level here that people call Rio base. And interestingly, the duration of the pulse, which has intensity 2i the Rio base, it corresponds approximately to 70% of the population membrane constant of the excited element. It's almost like a signature. And if you see basically what we get with the real data show that the chronaxy, as it is called, is about 220 microseconds. Very small chronaxes imply that the activation is basically restricted in a first phase in axons, and higher values, 800 or 1,000 or 1,200, it implies somata. And if you go to much higher, it means with whatever you're doing, you're stimulating the musculature. So basically, it's a good way to know approximately what is stimulated during the experiment. Now, we repeated all these experiments in area MT, and the results were the same. You can see here that basically the MT activates the PO, FST, the colliculus, and V1, V2, with the back projections of MT, and so on and so forth. What was strange, as I said, it was the following. On the left, you see the activation or the stimulation of V1, and on the right, you see the stimulation of MT. In the stimulation of V1, the stimulated areas are here, and they are all monosynaptically connected to the stimulated area. In area MT, the stimulated area, the activated areas are basically here, depicted here, and they are also monosynaptically connections. So this result basically raises the question, can electrostimulation induced excitation of cortical afferents propagating cortex? I don't need to convince you that that's an extremely important question, because when we do that, we imply that everything is going to be just like normal sensory stimulation. So it's important to know whether signals propagate in cortex the way the sensory stimulation propagates. And of course, to make sure that we can do that in a clean way, we stimulated LGN instead of stimulating the cortex. So everything is very clear, and you know what you're stimulating, and you have also certain expectations about the cortical signal propagation. So the LGN, I'm going to tell you, is a laminated structure with the parvo layers here and the magno layers here, and with some other very small cells that are intercalated zones that have not only projections to V1, but they have also strong projections to extrastriate cortex, in particular to the area MT. Most importantly, the LGN has a map. You can do the same mapping that you do with the stimulation of V1. There is very good retinotopy that you can exploit and go and first map the receptive fields and then know exactly where you're going to stimulate and where you expect to see the activation in the primary visual cortex in the area V2. This here summarizes basically the signal processing that you will have, whether you present something in the retina or you microstimulate the LGN at this particular location. You expect to have a small spot in V1 that will be activated, and then in V2. And as you see from the dashed lines, there are also some direct projections to extrastriate cortex, and there are more than what I did here. And there is one projection from, as I said, from the conjucellular system to MT. The slide shows the first results. The electrode is in the LGN, and you microstimulate the LGN, and of course this causes a positive bold response, as you would expect. A positive bold response is also seen in the superior colliculus and in the pulvinar. And of course a positive bold response is seen in V1, because as I told you, the Chronaxy and Rio-based measurements, they show that you're stimulating axons. The signal is guaranteed to reach V1. The thalamocortical, basically synapses, they will be active, and you will get stimulation of the primary visual cortex. What came as a surprise is what happens in V2. There is a very, very strong deactivation, and the same deactivation in this particular session you see in the area MT, and XC here is not a new name or a new area. It basically puts all the extrastriate areas together, so you know what's happening, because they behave exactly in the very same way. Now, as a note of caution, this deactivation is not the so-called sustained negative bold response that you might be aware of. This is pervasive in the literature. In every single experiment, particularly cognitive neuroscience, you're going to get red areas and blue areas. Whether you ignore them or not, this is your business, but you're going to get them. So there are deactivations. And you can see that here with the work we published a couple of years ago. Basically, you present a small stimulus, and if you present a small stimulus here parafoveally, then you're going to get close to the fovea, you'll get activation of the representation of this particular visual field location in cortex. And the areas that are distal, basically, to that, they are more peripheral. They have this blue color, which means they lost signal. And if you use stimuli that are of large eccentricities, then the opposite thing is going to happen. Now, keep in mind that these blue colors here, under the normal sensory stimulation conditions, they will always appear in areas that are not directly stimulated by the sensory stimulus. That's not the case with electrical stimulation. And you can better understand what I say by looking at the results with combined stimuli. Yep. No, you won't miss my beauty, don't worry. So, now what you see here is a design, an event-related design, where the green basically bar here shows the presentation of the visual stimuli, and the pink one shows the presentation of the electrical stimuli. They are randomly interleaved. And another design is you start basically with the visual stimulus, and 10 seconds later, you have a 10-second long presentation of the electrical stimulus on top of the visual stimulus, then the visual stimulus alone again, and then the baseline. And here are the results. Here now you don't see F statistics marked, but these are T statistics marked. Basically, the blue means the electrical stimulation is negative, and the red means the electrical stimulation is positive. You probably see that there is red in the LGN and there's the visual cortex. And the most important thing, if you see the time course, there is a positive response to the visual stimulation, and the very same areas that show the positive response, and this positive response to the visual stimulus is matching spatially also the electrical response, they will show the electrical positive responses in bold. On the other hand, if you see what is happening in the matching retinotopically matched basically V2 stimulation, the visual response, of course, it's going to cause a positive bold response, and the electrical response is going to generate a negative bold response. The same thing with combined stimuli. If you start with a visual stimulus in the primary visual cortex and in the subcortical centers, you get an additional basically increase of the signal, and in area V2 you're going to get a decrease of the signal that goes even beyond the deadline, beyond the baseline. Now this is not a problem of current intensity. Beyond the fact that the current intensities that we're using, they were used in a way that the current density, which is the critical parameter, was very low, lower than most of the studies, basically, that use microstimulation. We also tried very, very small currents, 10 microamps, 20 microamps, 30 microamps. No matter how much you reduce the current, what you're going to see is that there is a positive, basically, response in the V1 and a negative deactivation in the area V2. The left plot shows the relationship of the activated volume to the current, and as expected, you start with 100 microamps, and as you go towards 10 microamps, of course, the volume is reduced. The same trend is not evident in the extrastriate cortex. The curves that you see here were calculated by taking the time courses of the voxels that were active under all stimulation conditions. So basically, these are the voxels that always are active, and you see how they behave, and there is a dependency, of course, of the amplitude of the signal on the intensity of the current you use. The results are summarized here. These are the different locations that we used with 8 animals and many experiments. And basically, you see that for all the experiments, the lateral geniculate body, the colliculus and the pulvinar and the V1, they were positively activated. The V2 has a deactivation. MT has a positive, and the rest of the extrastriate cortex has a negative, basically, response. Now, this here is interesting because it was position dependent. It depended on where exactly the electrode is, probably because of the extent to which you are stimulating the coniocellular systems. Interesting is also the activation of colliculus and pulvinar. As you may know, with the exception of the reticular thalamic nucleus, the rest of the thalamic nuclei don't interconnect. So the colliculus, the LGN does not send a projection to colliculus and neither to pulvinar. So the reason you see this activation might be due to 3 possible things. The one is basically fibers of passage. The other is through basically collaterals that are coming from the retina, which are very, very weak. And the third one, the most likely one, and I think this is the case because of the data we have, is that basically the activation of the structure is due to the monosynaptic connections you have, the layer 6 of the cortex. And through the collaterals, yes, please. What about?
Yeah, the colliculus projections to the LGN and just your electrical stimulus.
If you don't have, yes, there would be a possibility if you had a method that can show antidromic stimulation. But MRI and 2DG and all the methods that rely on hemodynamic responses, they won't give any signal if you have pure antidromic stimulation without collaterals that project locally, because you don't have synaptic transmission. So this has been shown by many investigators and extensively by Sokolov and his group with very beautiful experiments using the 2DG technique. And they showed basically the only way you can get activation is you have synaptic transmission. Now, you could say maybe there are collaterals. To the best of my knowledge, and here we have a lot of specialists, there are no collaterals in colliculus or in pulvinar that would do exactly this thing. They get the signal in the antidromic direction, then they project it locally to the neighbors. The neighbors basically fire, and you have synaptic transmission, and you have a signal. Of course, the first thing we wanted to do, what is happening with physiology. And here you see on the top the rasters from the recordings, and as a response of a single pulse, and here you see the peristimulus histograms. Immediately after the short pulse, this is one single pulse with 200 microseconds duration. you have an immediate basic excitation that's probably due to the activation of the stellate neurons. And after that, there is a pause. There is a very large break that can be, you can see it here with a peristimulus histogram, that can be up to 100 milliseconds. The recovery time can be up to 500 milliseconds. So basically, the moment you send the pulse, you block the output of the recipient area. We applied simple clustering analysis to see what kind of responses you have if you take all the data you have into account. And there were two types of major responses. We did a lot of other things that I'm skipping right now, but this is the same result, whether you use principal component analysis, independent component analysis, or any other technique. And you see that the majority basically of the responses is this inhibition that recovers after 400 or 500 milliseconds. And these responses here are coming probably of the input layers of cortex. The positive responses on the right side, you see for the very same, indices of the clustering, you see the corresponding LFPs, and the thick lines are 0 to 40, or 1 to 40, and the thin lines are 60 to 100. That means the gamma range. So, and here is the distribution basically of the electrode tips that give the two type of responses. The blue corresponds to this inhibition here and tended to be more in the superficial layers, and the red signals, they were coming approximately from all layers. If you get the averages, these appear to be actually deeper than the ones that give this inhibitory response. What is interesting is the complete dissociation again of spiking activity in the supragranular layers and the LFP signals. Now, this behavior or the sequence of excitation and then inhibition with a slow recovery is something that people actually have reported already in intracellular recordings many years ago, but to the best of my knowledge, nobody so far has actually taken that into account for normal microstimulation and the propagation of the signals during microstimulation. This is a study by Kevin Martin and Ron Douglas, and they did intracellular recordings in the cortex of the cat and stimulated the afferent of the cortex. And what they noticed is there is a very short, basically, depolarization that you see here, and then there is a very quick hyperpolarization that was shown to be induced by the GABA A receptor, and a long-lasting hyperpolarization that was induced by the GABA B metabotropic receptor. The best explanation for this behavior, without going into the details, is not the typical perceptron neuron, the point neuron that integrates information, but this interactive, basically, excitation inhibition microcircuits that you see here, that explains the experimental data. It's a collection of both anatomical, reconstruction of buttons and everything data, and physiological data. And if you group the cells into the inhibitory neurons and the supra and infragranular glutamatergic neurons, then the principle of the microcircuit is a weak feed-forward input that goes to all of these cells, and a very, very strong recurrence. This strong inhibitory recurrence and the strong basic excitatory, the strong excitatory recurrence would immediately cause runaway excitation if the inhibitory neurons, they were not placed in strategic locations to very tightly control the excitation inhibition balance. And the moment they are activated, there is also an antagonistic relationship between these two groups. So the principles basically are weak feed-forward in inhibition, recurrence, long excitation inhibition evolution that goes beyond synaptic delays, and a driver modulation function of that. It can increase the conductances in parallel or anti-parallel. And in the first case, basically, you have no spiking, but you have changes in the activity of the whole area, something that, of course, you'll see also with fMRI. Now, what I wanted to tell you here is the characteristics of this microcircuitry here. They would predict that microstimulation that does not respect synaptic delays will never work in cortex. If you cause a spatiotemporal blur and you don't respect the synaptic delays, you engage the feed-forward inhibition way too early, and then you have also recurrent inhibition, and the signal is choked, and you don't have output from the stimulated areas. Also, because of this 100 milliseconds delay, you can imagine that there is going to be a frequency dependency, and indeed there is one. You can see here, basically, in V1, that there is excitation to visual stimulus. All the other things are electrical stimuli presented with different frequencies, 3 hertz, 6 hertz, 12 hertz, 24 hertz, and so on and so forth. You see that for something like between 5 and 20 or 30 hertz, even V1 is inhibited. It is inhibited because the inhibition is maximum, and the low frequency of stimulation does not generate enough thalamocortical activity to drive the bold signal. In V2, of course, there is also deactivation, and this deactivation has a minimum at about here, about 24 hertz, and this is exactly where you have the minimum activity also in V1. This change can be portrayed here by using standard, basically linear, the general linear model with the paradigm that I show you, with a visual stimulation and then electrical stimulation. And if you go to the V1, you'll see again that the visual stimulation is a positive response, and here is the electrical stimulation positive response. And in V2, you're going to see positive response here and negative response here for 200 hertz. But the moment you reduce the frequency, you go to 8 hertz, both V1 and V2, they will be deactivated. Now, we wanted to understand that, and we did extensive physiological studies simultaneously with fMRI or separately using arrays of electrodes. And here is a very brief summary of the data. And we also used actually GABA blockers to make sure that indeed the effects that we see are due to the fact that you have a feed-forward inhibition shutting down the output of the area. On the left here, you see spatial independent component analysis. What that means, basically, you try to understand the statistics over time of a particular set of block cells and see which kind of transformation generates groups that are maximally, from an information theoretical point of view, maximally independent from one another. And these were selected in an unsupervised manner. There is no model, there is nothing here. The different areas that you see here that are spatially filtered were areas where the time course of the signal was distinctly different in the region here, was distinctly different from this region, and so on and so forth. Now, if you see the time courses of these regions, you see that there are characteristic profiles. The one here shown in blue, and is coming around the electrode, shows a very strong baseline shift because of the application of the GABA blocker. The GABA blocker is applied where the dashed line is. The ups and downs, they are modulation induced because of the electrical stimulation. It's 32 seconds on stimulation, 32 seconds off. So you have basically, every time you have on, you have basically modulation of the bold signal. And on top of it, you have a very large baseline shift. As you go far away from the tip of the electrode, you'll see only the modulation of the electrical stimulus without seeing any baseline shift. The green area is the very interesting area. I called it here IPZ, is basically the injection projection zone. This is the retinotopical image area that receives information from that primary visual cortex region that received the injection. And it has basically a drop in the activity. And most importantly, you can see it here, if you average the trial, you'll see that this particular region is initially negative, the way you would expect it from the data I showed you previously. And after the injection of the gamma blocker, there is a clear positive response. The thick lines here are after the injection, and the thin lines are before the injection. In V1, you would expect it to be the same before and after. And in area V2 that is far away from the injection site, the signal polarity remains the same. You have basically inhibition. But in the area that receives the effects basically of the gamma blocker, you have an inversion of the signal, demonstrating that indeed it is the inhibition that shuts down the output of V1, and the loss of the output of V1 for V2 causes the deactivation of the latter area. This here summarizes all the data for several sessions to show you how robust these kind of results are. Here I have indicated the different basically regions that have distinct time courses. These here are selected again in an unsupervised way. So this area and this area, they have time courses that are distinctly different. So if you see the time courses in the far basically region, far with respect to the tip of the injector, in the far region of the primary visual cortex, you'll see the electrical stimulus-induced basically modulation, and there is no baseline shift, and there is no inversion of the signal. And in the far area of V2, there is going to be an anti-correlation because every stimulus is causing the activation, and there is no other change in the signal. On the other hand, if you see what's happening around the tip of the electrode, there is this baseline shift. And if you see what is happening in the recipient zone, forget the details here, there is an inversion, and I show you that is summarized here. The before the injection trace is the blue one, and the after injection trace is the red one. And there is a very clear inversion, basically the activation now is a clear positive bold response. Now, I mentioned in the beginning that you don't have only the sequential pathways, but you have also direct pathways. That means you have V2 fibers going to, LGN fibers going directly to V2. And the question that we wanted to ask is, in fact, actually we did work recently, and you show that you can do very good retinotopic mapping, despite of the fact that you don't have the V1, and we did it by using basically animals that had V1 lesions. Now, the question we want to ask here, if indeed you have lesions and the V1 output to V2 is blocked, then is this enough to cause a positive bold response during electrical stimulation? And here is basically the experiment. You see that there is a retinotopic mapping, as you would expect. And this is the lesion area. This is the V1 lesion here shown with blue. And this is the intact V1. The V2 lesion projection zone is within this red, basically, contour. And this is the intact V2, the control. And the circle here shows the mapping that you can do. Indeed, you can do mapping within the lesion projection zone, like the mapping you can do with the intact cortex, except that the signals are weaker. If you now measure the bold response during electrical stimulation, in the control V2 area, you're going to see what I showed you already. The visual stimulation is going to cause an increase of the bold response, and adding the stimulus is going to cause a very deep deactivation of the area. If you see what is happening in the lesion projection zone, where you only have the direct input from the LGN, then you see that there is a very clear positive response. Now, you don't have any loss because there is no V1 output to this area to begin with. So activation maps obtained by pulvinar stimulation, on the other hand, they look completely different because, as I told you, pulvinar tends actually to project to all these areas, not in a sequential manner. And the results show basically that stimulation of the pulvinar is going to cause a positive bold response in all the subcortical structures and also in area V1, MT, and XC. And this is also verified with a very large number of experiments and 7 or 8 animals. The conclusions from these cortical and thalamic stimulations are here. And the very first one says that the complexity of the cortical microcircuits and the strong recurrence suggest that optimal operation of these units can only be warranted for spatiotemporally structured input patterns that respect synaptic delays. During electrical stimulation, the unnaturally over-synchronized excitation of cortical afferents activates both excitatory and inhibitory pathways that shunt the spiking of cortical neurons. Shunting is due to the spatiotemporal blurring rather to the stimulation stimulus intensity itself. Now, I told you from the beginning that these results are not only important for basic research that's in my interest, but they have also important implications for translational research and for clinical research. And I wanted to show you here a quite impressive, actually, video, and we have it also in the network, that shows the effect of deep brain stimulation on the patient and explain you a few things. First, I'll show you, if I can manage to, okay. So, oh, that's very bad. Okay, it doesn't matter. So, it doesn't matter. So what it shows basically that the patient is, that's very nice. I should go to sleep. What it shows is the patient is is at the very late stages of Parkinson, and he's really shaking. His whole body is shaking, and They put this, he stops, he can talk, he can explain things, he doesn't have the tremor. And then he disconnects himself from the pacemaker, the brain pacemaker. He starts, of course, the Parkinson, and he connects it again. So it's a spectacular, basically, treatment of the disease, as you all of you know very well. The thing is that about 40% of these patients, they have all kind of adverse reactions after the first 6 months. Some of them, they get schizophrenia. Some of them, they get other things, and both neurological disease and other things. And the reason for that is because there are really certain things that have to be examined finally in detail. One of them is the exact extent and shape of the directly activated brain area, as I told you. And it is like to be area and electrode dependent. It depends on the horizontal connectivity, on the lateral connectivity, on the collaterals, and so on and so forth. And relative contribution of different elements, dendrites, somata, and axons, to the direct activation of neurons. The conclusion that we draw, including the one I drew myself here, that electrically evoked activity in gray matters is attributed mainly to excitability of axonal elements is probably misleading because of the recurrence and the intrinsic axon. That is, even when only a small number of neuronal somata have been stimulated directly, a large number of dendrites somata will be activated by antidromic stimulations if you have collaterals that project to the same area. Finally, technology, spatiotemporal variation of current density, probably, might be needed to minimize the spatiotemporal blurring that causes this faulty, basically, activation of the microcircuits. So, the results I presented to you, they have also certain implications for both electrical stimulation, as I said, and the TMS. Because both the ES and the TMS are likely to silence all cortical areas receiving direct input from the fibers they excite. The result of ES-TMS may need to be interpreted as showing what an area does by itself together with all the subcortical projection it has, or what kind of behaviors follow the reduction of activity in the monosynaptic targets of the stimulated region. A very last thing, the activation of P1, despite the strong reduction of the area's output, the spiking, as well as the inactivation of V2 as a result of V1 input loss, once again demonstrates the very high sensitivity of the bolt signal to input and intracortical processing. I'll now continue the last part of the talk. Do I have the time, or it's over? If it's over, I can stop. 5 minutes, okay. With the hippocampus stimulation. Well, I don't need to tell you much because I know people are aware of what LTP is and what hippocampus is doing. It's a beloved structure of anybody that's interested in spatial navigation and memory and so on and so forth, and has been very tightly linked with the long-term potentiation. People did a lot of studies over the last years, and we know a lot of local details. Here you see the diagram, basically, that was done by Cajal, and here you see an easy version of it. You have basically the perforant path that projects to the dentate, and from there you go to CA3, CA2, CA1, and then subiculum, and then back to entorhinal cortex. You have indirect pathways and direct pathways. What we wanted to do is to stimulate there under conditions that you can induce LTP and see what is happening in cortex. The image here is 100 micrometers isotropic scan. It's without any contrast agents. It shows very good contrast. You can see all the structures. And on the left side, you see basically the stimulation electrode, and on the right side, you see the recording electrode. This is now a combination again of intracortical recordings, electrical stimulation, and fMRI. And this is the typical responses you get. It's not different from cortex. You're going to get the positive bold responses. If you stimulate the perforant path, you're going to get the negative bold responses. And the characteristics, the dynamic characteristics of the responses, they are pretty much identical to the ones you could get if you stimulate any other structure. And you see here the activation, basically, of the dentate gyrus. And I have here the atlas for those of you that may not be remembering the subfields of this structure. Now, the activation is, as expected, frequency and intensity dependent because of these direct and indirect projections of the perforant path. And you see here that the moment you go to the higher frequencies, you have also commissural pathways activated. So you activate the other side, basically, of the hippocampus. The very first thing we did, we selected a low-frequency protocol that ensures that you're not going to have any changes in the responses when you use this protocol for several hours. You see here stimulation with 250 microamps, very low frequency, and stimulation with 750 microamps if you use. There's a temporal stability. This is here the population spike, and this here is the EPSP, and this is the bolt. They are all stable. You can, for many hours, actually stimulate in this way. No changes are going to happen. Then we went and we applied the LTP protocol. Just in case that somebody here is not familiar with the protocol, you apply high-frequency stimulation. There are other tricks. And with this high-frequency stimulation, you induce basically long-term potentiation. It's a plasticity basic experiment. And after that, you go back to very low frequency stimulation. And now the responses are completely different. And this is shown here. This is the population spike. I remind you, this is has nothing to do basically with the number of the firing rate, or has nothing to do with the amplitude of the spike. It has to do with how many neurons are participating in the activation. And you have basically the EPSP, and after that you have the inverted here portion of the curve that reflects the population spike in the dentate. Now, if you have the induction phase and you induce LTP, then you have two changes that people have reported for several years. Is the slope of the EPSP, and is also the amplitude of the population spike. Both of them are strongly basically altered. And here is the time course. Here is continuous low frequency stimulation. There is induction here, and you continue with the same low frequency stimulation, but you're going to see now this low frequency stimulation causes a lot of increase in there is potentiation in the population spike amplitude, which goes down, and the returning to the initial value may take from 2 to 3 days to 72 days. I think it's the longest people have reported in the literature. So we wanted to repeat this experiment with Bolt, see first of all it works, and then the most important thing, see what happens to the rest of the brain, something that you cannot address with traditional physiology. Here is basically stimulation of the perforant path. And you see that clearly the Bolt after the induction of the LTP is higher. And if you see what's happening basically with the time course of the Bolt amplitude, you'll see that there is a potentiation, the way you saw previously for the population spike, and then it goes down, exponentially goes down to the baseline. And the population spike, which is superimposed, shows pretty much the same time course. And here are the differences between the amplitudes. Now the question, of course, is this an MDA dependent? And we actually used the MK801 injection, and you see that before the injection, after the injection, there's a small drop. But whether basically you have, if you block the NMDA receptor before actually the LTP and after the LTP, the amplitude of the signal remains the same, which means, as you expect in DG, you have an NMDA-dependent LTP, and you'll stop it if you inject an NMDA blocker. And the final slide here answers the question I asked you, which can only be actually asked with these combined methodologies. Here you have the activation basically of the hippocampal formation of the dentate, and after basically the LTP, as I told you several times now, you have an enhancement in the Bolt responses. But what was much more interesting is what is happening in the rest of the brain for exactly the same low frequency stimulation. You see that suddenly several other areas kick out. This is the perirhinal cortex, is accumbens, is prefrontal cortex that were not activated before. They are activated after you change the basically synaptic strength locally in hippocampus. So, and if you use again an MDA blockers, then you're not going to see any changes in the cortex. This is work that we published recently. So the LTP induction changes the Bolt response of the target subfield of the hippocampus. LTP induction results in network reorganization that includes increased interhemispheric communication, I didn't have time to show you, and the recruitment of limbic and neocortical circuits. The nature of the recruited structures points to an increased communication between associational polysensory cortices with the cortical and subcortical limbic networks subserving memory. The number of targets of the hippocampal output greatly exceeds the number of extrahippocampal structures shown in Bolt maps in these studies, suggesting that the functional consequences of synaptic potentiation do not uniformly affect the entire anatomically defined network. And here I would like to stop and thank you for your attention.
I think we can take about 3 or 4 questions. Anyone have any questions? Go ahead.
So I find your LTP results remarkable, and I'm curious if you tried LTD.
No, we didn't. I should say that the data I'm presenting with the hippocampus is really new work. It was published because it was very robust results that we did, I think, 25, 26 rats, and it's believable, and we publish it, but we are not going to publish anything before we work quite a bit with that, because there are a lot of details. There was the first observation, is very robust. If you do that, you're going to get the same results, and the activated areas that are engaged are going to be the same. They're going to be the same as long as you induce LTP indentate. As you know, you can induce LTP in CA3 or CA1, and the results are expected to be different because there are different mechanisms of LTP. So we don't know what the answers are. We're starting now, and in fact, actually, we are very much interested to move to the monkey because there's pretty much nothing there, and we want, and I feel much more comfortable with the rat brain. I have to read 300 times the atlas to see where the hell I am, and if I have the monkey brain, I know where I am. So go ahead.
What would the human homologue be to the prefrontal cortex and the other remote area, ACV? Was that cingulate lung volume?
Accumbens. Accumbens. Yeah, nucleus accumbens.
What would the prefrontal cortex homologue be from rat to human, if you had to guess?
I do not know. I do not know. Yeah. Yeah. Actually, I should say these studies will continue. We won't even continue them with electrical stimulation, but we are setting up right now for optical stimulation. Optical stimulation and optogenetics seems to be a good way to do these things. They are also artifact-free. Of course, the translation value of that is close to 0 because it's going to be quite a bit of time before someone dares to apply all these genetical manipulations.
Fascinating in the monkey to see if you get ventrolateral prefrontal cortex stimulation.
Correct. Yeah. Correct. With monkey, it's a completely different story, and we can better basically predict what should be happening and also test it. With rats, I'm relying on whatever I can find in the literature with relatively superficial reading because I'm not familiar with the rat brain. But okay, I mean these experiments were simple. They were simple. There was nothing. Yeah, go ahead, Ken.
So if you can only bring positive signals from microstimulation one structure away from your where you stimulate, how do you explain all the systematic positive effects on behavior? Are you suggesting then that one synapse is all you need to get to perception or a decision?
You can say that as well. I do not know, but as I said, you have these loops that you don't block. The microcircuits are extremely blocked in the supragranular layers. The layer 6 neurons, they will project back, and the layer 5 neurons, they may project back. And as you know, the LGN sends information actually to B1, then with layer 5, with the driver connections to ionotropic connections, you go to pulvinar, and then with the driver connections, you go to V2. That makes this replication principle. So there are other pathways. My point is not that there is a single study out there that reported anything false or anything. It's absolutely not. It's a joke. In all the studies, whatever they reported, it's the way they reported it. But if you try to tell me how you think it happened, then we'll have disagreement. You know? Whatever you got, you got. I mean, you get it, and yeah. So it's basically, and then we have also physiological evidence, a really, really very robust result, and in many ways also, to be quite honest, if you read the literature, you can predict that. But this information was not used. That part is definitely new in this context, because David Ferster, for example, and Clay Reed, they were trying to map the physiological properties of input neurons. And in order for them to claim they receive an input that has such a spatial organization that you have orientation selectivity already at the level of the stellate, the microcircuit has nothing to do with the orientation selectivity in primary visual cortex. And that basically justifies all the claims of Hubel and Wiesel and so on and so forth. To try to do this experiment, what they did, they were trying to silence the microcircuit stimulation. And if you think of it, on one hand, you do this trick to silence the microcircuit. On the other hand, you want to have prosthetic devices that are supposed to do something. They may do still, but this is only because everything is so opportunistic in the brain. I mean, you never lose. You never win either.
I'll take one more question.
Given the fundamentally different organization of the cortex and the hippocampus, I was curious why you would expect the same pattern of fold response in both structures.
Why do you
Why
expect
you
what?
would get the same pattern of results given the different organization of the two types of cortex.
Why you get activation and deactivation?
Yeah.
Because you still have basically feedback, lateral connections, everything. And this, the activation that I was showing with hippocampus is basically like just the deactivation. You see when you use, in the sensory area, if you use a sensory stimulus, there are always negative bold and positive bold areas. You get that as well. And I was excluding it by concentrating only in the areas that show basically positive responses during sensory stimulation. So when you do fMRI, for good or for bad, you don't assume that every single thing that you see should have a different interpretation. Until we come up with such a conclusion derived from experiments, we are assuming basically the basic responses, they're going to be the same from structure to structure. The magnitude will change, for sure, because also the vascular density varies very much, and the hemodynamic response functions are different. But you don't expect, actually. So we have no evidence that we should be expecting completely different response patterns.
Okay, I think that's all the questions we can take. Let's thank Dr. Logothetis for a truly great presentation.